Hi students, welcome to SPSVERSITY. In the previous session, we discussed about the principles of inheritance and variations. So in this session, we are going to discuss about the molecular basis of inheritance. So what is meant by molecular basis of inheritance? So in Mendel's experiment, we studied that genes and chromosomes play an important role. What are these genes and chromosomes are made up of? So what molecules will play an important role in inheritance? That is, genes and chromosomes are made up of a macromolecule, that is the DNA. So here when we come to a biomacromolecule, that is, they are called nucleic acids, which play an important role in inheritance. So nucleic acids are the macromolecules present in all living cells, whether it is a microorganism or whether it is a complex organism, all the organisms have nucleic acids. So the first nucleic acids were isolated by a person called Frederick Michel, and he isolated from pus cells. That is, when any injury occurs, a pus is formed in the part where injury took. So from the nucleus of pus cells, it is extracted. That's why it is called nuclein. So why it is called nuclein? The reason is it is extracted from nucleus of pus cells, and they show acidic nature. That's why they are called nucleic acids. So Altman called this as nucleic acids. The reason is they are acidic in nature. So basically nucleic acids are acidic in nature. So that's the reason why they are taken from, that is, they are called nucleic acids. As they are called nucleic acids, that is, as they have acidic properties, so they are called, that is, as they show acidic properties, so they are called, that is, they are, as they show acidic properties, so they are called nucleic acids. So there are basically two types of nucleic acids found in organisms. That is, deoxyribonucleic acid, one is called DNA, and another one is called ribonucleic acid. What is this DNA and what is this ribonucleic acid? So DNA is a molecule that carries genetic information. So genetic information means whatever the information present is, that is the blueprint in for living cells, it is present in nucleic acid molecule, that is the deoxyribonucleic acid. And this deoxyribonucleic acid, what it is made up of? So the structural units of the deoxyribonucleic acid, they are called deoxyribonucleotides. It is deoxyribonucleotides. So the basic structural unit of the DNA is deoxyribonucleic acid nucleotides. So this deoxyribonucleotides, that is, a pair of deoxyribonucleotides is called base pair. Like this, depending upon the species, the number of deoxyribonucleotides will be specific, which decides the length of DNA. The length of DNA depends on the number of base pairs. So depending upon the number of base pairs, the length of the DNA is dependent. So if we come to the examples, like for example, if we take bacteriophage. So bacteriophage pi into 174, it has 5386 base pairs. And when you come to lambda bacteriophage, it has 48,502 base pairs. Then you come to bacteria Escherichia coli. So Escherichia coli has 4.6 into 10 to the power of 6 base pairs. And when you come to humans, that is the haploid number of, that is when you take the haploid cell of a human being, it has 3.3 into 10 to the power of 9 base pairs. So the length of the DNA is not same in all organisms. It varies from one organism to one species to other species, depending upon the length of, that is number of base pairs. So when you come to this base pairs, that is when you come to the structure of DNA, as we already discussed, DNA is made up of deoxyribonucleotides, which are joined by covalent bonds. So here when you come to nucleic acids, that is the nucleic acids, they are made up of the micromolecules, that is nucleotides. So nucleotides will come under, that is micromolecules, which form a macromolecule, that is the nucleic acid DNA. What these nucleotides are made up of? So nucleotides is made up of three basic components. The first one, it is made up of a sugar, that is a pentose sugar, deoxyribose sugar. As it is having deoxyribose sugar, that's the reason why the name deoxyribonucleic acid has come. So the name deoxyribonucleic acid has come based on the presence of a pentose sugar, that is deoxyribose sugar, and a phosphate group, and next a nitrogenous base. So in these three components, there are no subtypes in deoxyribonucleic sugar and phosphate. But when you come to a nitrogen base, there are again subtypes in nitrogenous base. So in nucleotides, the types of nucleotides will be due to the nitrogenous base. What are these nitrogenous bases? There are, based on their structure, there are two kinds of nitrogen containing bases. So one is called purines, another one is called pyrimidines. So purines have double cyclic ring structure. So they have double cyclic ring structure. In this, one ring has 6 positions and another one has 5 members along with nitrogen. So there are 2 rings, one with 6, another one is 5 members along with nitrogen. So here the nitrogenous bases are made up of carbon along with nitrogen. Whereas when you come to pyrimidines, it has only monocyclic ring structure. Mono means only one ring like structure is present. It has 6 members with nitrogen. And again in purines, again there are subtypes. So finally, if you take the total number of nitrogen bases, that is, they are adenine, guanine, thymine, cytosine, and uracil. A, G, T, C. A and G come under purines. P, C, U, thymine, cytosine, uracil, they come under pyrimids. So finally, there are 5 types of nitrogenous bases. So these 5 types of nitrogenous bases will constitute, that is, will form a nucleotide. So in the formation of nucleotide, that is, if you take a structure of a nucleotide, so what you are seeing, it is a nucleotide. This is a nitrogen base, this is a pentose sugar, and this is a phosphate. The bond present between, that is, the bond present between nitrogen base and sugar is the N-glycosidic linkage. Is N-glycosidic linkage. And the bond present between the sugar and phosphate is phosphoester linkage. Sugar and phosphate are linked by phosphoester linkage. Here in the formation of the nucleotide, initially nitrogen base forms a bond with sugar. So this nitrogen base along with sugar, it is called nucleoside. And when it attaches with phosphate, this one is combinedly called a nucleotide. So here, in the formation of a nucleotide, initially nitrogen base combines with, that is, it forms a bond with ribose sugar, that it forms a bond with pentose sugar by glycosidic linkage. Later on, it forms this one strand. As it is made up of 2 strands, it is called double-stranded. So if you take the double-stranded, it is called DS, DNA. In most of the DNA, there are 2 strands, and each strand is made up of many nucleotides. So each strand is made up of many nucleotides. So these are the nucleotides of one strand, and these are the nucleotides of one strand. And here, the nucleotides of one strand and the nucleotides of other strand are linked by hydrogen bonds. So these are hydrogen bonds. And there is a specific formation of hydrogen bonds between the nitrogen bases of a base pair. For example, if you take this, the nucleotide of one strand and the nucleotide of another strand, which are opposite each other, they are called base pair. It is called nucleotide base pair. So in a base pair, there are 2 nucleotides. One is, that is, one which belongs to one strand, another one belongs to another strand. And the 2 nucleotides are linked by forming a hydrogen bond between the nitrogen bases. So here you can see 2 hydrogen bonds are formed between adenine and thymine. Here 3 hydrogen bonds are formed between guanine and cytosine. So here if you take, every DNA and RNA molecule has a direction. That is, it is called 5N to 3N. Any direction if you take, it is represented by 5N. One end is called 5N, another one is called 3N. Okay. So while you are writing the sequence of this, you will be writing like this. That is, the 5N If you take the 5N, we'll be writing at one of them, and 3N where that is the OH group of the sugar molecule is free. That's why we'll be writing 3OH group. Whereas the fifth group of the nitrogen base is free. That's why we'll be writing as 5N. Okay. So here when we come to DNA specifically, as in DNA there are two strands and always bond is formed between, that is always a bond is formed between A, T and G, C. That is between A and T, 2 hydrogen bonds, between G and C, 3 hydrogen bonds are formed. So this relationship where there is a complementary relationship here, A always binds with T, G always binds with C. As it always binds with C, the proportion of purines and pyrimidines, adenine and guanine will come under purines, thymine and cytosine will come under pyrimidines. So the ratio will be always equal, that is 1:1. That means if there are 10 adenines, there will be 10 thymines. If there are 40 guanines, there will be 40 cytosines. So what Chargaff observation says is the ratio of purines and pyrimidines will be always 1:1, okay, as they are showing complementary relationship. Okay. To find out the structure of DNA, so many people struggled with a lot of experiments on, like for example, if we take Franklin, a person who has done X-ray diffraction experiment. He has done an X-ray diffraction pattern DNA. And in this X-ray diffraction of DNA, he has done analysis of DNA. After doing analysis of DNA, based on the Franklin X-ray diffraction method, Watson and Cricks proposed the double helix model of DNA. Why it is called double helix? Two strands are there which will be intertwined with each other. So this is one strand, this is one strand. So here, if you take the strands, one complete 360 degrees, one turn is 360 degrees, it is called, its measurement is 3.4 nanometers. And there are, the distance between two base pairs is 0.34 nanometers. And the distance between two strands is 2 nanometers. So here, based on Franklin X-ray diffraction method, so this Watson and Cricks proposed the double helix model of DNA. So when you take double helix model of DNA, in a DNA there are two strands which are anti-parallel. Anti-parallel means one will be in 5 to 3 direction and another will be 3 to 5 direction, which will be nucleotides. And in this, each strand, the backbone is formed of sugar and phosphate. And the two strands are anti-parallel. Two strands are anti-parallel means they will be moving in opposite direction. So 3 to 5, 5 to 3. Okay. And here adenine always pairs with thymine by 2 hydrogen bonds. Guanine always pairs with cytosine by 3 hydrogen bonds. And the two chains are coiled in right-handed fashion. They show right-handed coiling. So they show right-handed coiling. And when you come to the parameters of DNA, one complete turn, that is called one pitch or helix, the length is 3.4 nanometers. Or if you write in this, That is, if you write in Armstrongs, it will be 34 Armstrongs. And the distance between two base pairs is 0.34 nanometers. It will be, when you take in angstroms, it will become 3.4 Armstrongs. And the distance between two strands is 2 nanometers, that is 20 Armstrongs if you write in Armstrongs. So these are the parameters of DNA. And when you come to DNA, so this DNA plays an important role in flow of genetic material. This plays an important role in the flow of genetic information. What genetic information? In what form? Like DNA will form RNA, RNA from RNA protein is formed. So DNA plays an important role. So till now, what we discussed is the structure of DNA, where it has two polynucleotide strands, where they are linked by the hydrogen bonds, the base pairs, and the nucleotides are joined by, that is, by the help of hydrogen bond between nitrogen bases. Okay. And when you take the length of DNA, so the DNA is very long. It's like a long chain. Such a long chain of DNA, how it is packed in cells. Cells are very small. In such a small cell, long chain of DNA is present. How it is possible? Is it possible? Or what is the mechanism involved in this? How they are packed? First we'll see in prokaryotes. How DNA is packed in prokaryotes. In prokaryotes, they don't have a definite nucleus. And even though it doesn't have definite nucleus, the DNA is not scattered. It is held together with some proteins in a specific region that is called nucleoid. So nucleoid means the region in which the DNA are held together with proteins. The DNA in nucleoid is present along with proteins is present in the form of large nucleus. They come to packing of DNA in eukaryotes, where it is still more complex than prokaryotes. In prokaryotes, there is no nucleus, but in eukaryotes, there is a nucleus. So in a small cell, inside the nucleus, the DNA is present. How it is packed? So when you come to eukaryotes, when you take specifically, so eukaryotes, in eukaryote, DNA is associated with a basic histone proteins called histone proteins. That is, DNA is associated with positively charged protein, that is, they are called specifically histone proteins. Why they are called histone proteins? What is the reason? What is this histone proteins? They are rich in basic amino acids. That is, they are rich in lysine and arginine. Histones are rich in lysine and arginine. And these histones form a histone octamer. That is, when you come to histone proteins, in histone proteins, if you take, so in histone proteins, there are H1, H2A, H2B, H3, H4. There are 5 types of histone proteins: H1, H2A, H2B, H3, H4. So 2 molecules of H2A, 2 molecules of H2B, 2 molecules of H3, 2 molecules of H4. That means total H2A, H2B, H3, H4. Total 8 molecules of histone will combine and form an octamer. Octa means 8. See the figure what you are seeing, that is what you are seeing in the center, that is a core, this is called core of histone molecule, which is formed of 8 molecules of histone proteins, which is called histone octamer. Surrounding to this histone octamer, the DNA coils for twice. This DNA which coils for twice. And the ends of DNA are held by H1 histone protein. The ends of DNA are held by H1 histone protein. So here what you are seeing, that is each one is an, that is here you are seeing. Each one you are seeing and it is associated with histones. So this is the chromatin material, nothing but DNA, which is present in the form of like this, which is associated with the protein. This is the DNA chromatin, which is associated with histone proteins. Okay. So this, along with histone proteins, the DNA is associated. So here you can see, this is one octamer, this is one octamer. So here you can see there is these are closely packed where the genes are inactive. Here they are loose where the genes are active. Okay. So here this is a chromosome which is made up of the DNA. This is the gene which is, this is the chromosome which is made up of DNA. Okay. So this entire structure where it is associated with 8 molecules of histone proteins along with DNA, this entire structure is called nucleosome. So such like in DNA there are many nucleosomes. So the DNA is negatively charged. It wraps around the positively charged histone proteins to form a nucleosome. And nucleosomes looks like a beads on string. And this nucleosome forms the repeating units of structure of chromatin. This is the entire chromatin where there are many repeating nucleosomes. Each one is one nucleosome. So this is H1 histone protein. This is DNA. This is octamer. So like this, we can see the chromatin. Okay. So chromatin is nothing but DNA, which is the repeating units of, with DNA with repeating units of nucleosome, and it is coiled. Okay. So this chromatin, it forms along with further coiling and condensed, it forms, that is, chromosomes. Okay. As we already discussed, in the nucleus, in some regions, the nucleosomes, that is, which are loosely coiled, the regions of DNA which is loosely coiled, it takes less stain, that is called euchromatin, which has active genes. That is, it is active transcriptionally. Heterochromatin, what is this? Where it is densely packed, it takes more stain, it is called heterochromatin. That is, it is loosely packed, whereas euchromatin is loosely packed, whereas heterochromatin is tightly packed. Euchromatin is transcriptionally active, whereas heterochromatin is transcriptionally inactive. So what is meant by euchromatin? Where the DNA is coiled loosely, whereas heterochromatin is DNA is packed tightly, which is transcriptionally inactive than euchromatin. Okay. So we are saying genetic material. DNA is a genetic material. What are the properties it is having to satisfy the genetic material? So DNA can replicate on its own. That is, from one DNA on replication, it will form another DNA. It is structurally stable. It can undergo changes, mutation, and it is able to express itself. That is, it is able to show Mendelian character. Based on this four criteria, the DNA is confirmed as a genetic material. Now we'll see what is the difference between DNA and RNA. The DNA is called, full form is deoxyribonucleic acid. RNA is ribonucleic acid. It has deoxyribose sugar, where it has ribose sugar. Okay. Phosphate both have. But here they have adenine, guanine, cytosine, and thymine. No uracil here. So in DNA there is no uracil nitrogen base. But here adenine, guanine, cytosine, uracil. So no thymine here. Here thymine is absent. DNA can replicate, but RNA cannot replicate on its own. It is double-stranded, whereas hence most it is single-stranded. It doesn't have any catalytic role. That means it doesn't show any rules like enzymatic rules. But RNA plays an enzyme, that is, it acts like a ribozyme. That is, it acts like a riboenzyme during protein synthesis. Okay, till now we studied about the structure of DNA and its properties, the characteristic features, the structure, the double helix. Okay. On what basis, that is how they find out the genetic material, that is the search for genetic material. How they have searched for this genetic material. What are the experiments? So the experiments, that is one of the first experiment is conducted by Frederick Griffith. Okay. It is saying the bacteria are capable, that is, he has conducted in bacteria. Okay. According to Frederick Griffith, the bacteria are capable of transferring genetic material information through a process known as transformation. So how he has reached this conclusion? So for his experiments, he has taken a bacteria, that is Streptococcus pneumonia. So which causes pneumonia? It is a Streptococcus pneumonia which will cause pneumonia. And Griffith has used two strains of Streptococcus pneumonia. One is smooth strain, another one is rough strain.